Hello everyone, welcome. So today what we're going to do is we're going to cover 5B. So in 5A we looked at photosynthesis in general, how it functions in three plants, what the inputs and what the outputs were and what the purpose of it was. What we're going to do today is we're going to look at light independent stage in a little bit more detail. We're going to do that now because it's going to link in to looking at adaptations between plants and how different types of plants like C4 and CAN plants have changed their photosynthesis slightly to encourage rubisco in its job, in its light independent reaction. Let's just say that. So before we start, this is just a quick reminder of what we did in our last video. So we looked at the light independent reactions, and the light independent reactions are also called the Calvin cycle sometimes. So you might see it written as that. So our light dependent reactions, they occur in our grana, in our chloroplast. So the light obviously is absorbed by the leaves through chlorophyll, and this light causes hydrolysis or photolysis, which is the splitting of our water input. So this water is going to split into oxygen, which is a product which is released outside of the plant because we don't really need it, and it's also going to be split into hydrogen ions. This is really important because this splitting and this creation of hydrogen ions does two things. So these two things is that it powers ATP synthase to convert ADP into ATP, which is really important for our second stage, and it also converts NADP into NADPH. So because it adds on the H plus ion. So the main purpose of the light dependent reaction is to load up our coenzymes, which is ATP and NADPH. So the main focus is to load them up into their loaded forms so that they can assist in the next cycle. So the light independent stage is where we have our input of carbon dioxide, which we take in through the stomata, and this carbon dioxide undergoes something called the Calvin cycle or carbon fixation, which we're going to look at in a bit more detail today. So basically these coenzymes here, ATP and NADPH, that were used, which were created in the last stage, are used in this carbon fixation process, and the main goal of this process is to convert inorganic carbon in the CO2 into organic carbon that can be used to create our glucose molecule. And as a result, glucose is a product. So glucose is a product, and we also have a little bit of water as well. So that's just a refresher of what we learned about photosynthesis. The main goal, of course, to produce that glucose molecule for energy. What we're going to do today is we're going to learn about the light independent stage in a little bit more detail, and we're going to learn about this enzyme called rubisco. So I alluded to this in our last video a little bit. Rubisco is the main enzyme that is involved in the light independent stage. So rubisco is responsible for this carbon fixation process that I've been talking about. So its main role is to turn carbon dioxide into an organic form of carbon that can be used to produce sugar. So, of course, as a result, the main substrate for rubisco is carbon dioxide. So carbon dioxide and rubisco are going to bind, and this is what facilitates that process in photosynthesis. However, rubisco is remarkably inefficient. It is a stupid enzyme. Honestly, I will say it. It is a stupid enzyme because it's got a lot of problems with it. It's remarkably inefficient. It only does 3 reactions a second, which you might think is really good, but it's actually not very good in terms of enzymes. So to compensate, it makes up almost half the protein content in chloroplasts. So that's one problem with rubisco. There's also another problem with rubisco, which we'll go through a little bit later on, which is called photorespiration. So rubisco is very silly. It doesn't know what it wants to bind to. It's very indecisive, and it initiates this wasteful process that we're going to talk about a little bit later on. But for now, rubisco is the enzyme that, in perfect conditions, is going to facilitate this carbon fixation process in the light independent reactions. So what is this carbon fixation process? So this diagram probably looks a little bit alarming, but you don't need to know every single little itty bitty bit of it, but all very good to know because it might well come up in an exam or a SAC. So essentially what carbon fixation is, or this Calvin cycle that happens in this light-independent reaction, is the cycling around between carbon molecules to produce glucose. So we know that this uses CO2 and it uses the coenzymes that are made in the light-dependent stage, NADPH and ATP. So because plants can't convert carbon dioxide directly into glucose because it's wasteful of energy, so it takes way too much energy to do that, it goes through this cycling process. So this cycling allows CO2 to create an organic form of carbon to be made into glucose. And rubisco is the main enzyme involved in this. So rubisco is going to bind to the carbon dioxide and it's going to take it through this carbon fixation and this Calvin cycle process. So it can be split up into three main stages, which I've sort of summarized to hopefully make this diagram a little bit less daunting. So the first stage is carbon fixation, which happens in this little piece of the pie here. So this is mainly what it's the main thing that you need to know. The last two stages are good to know, but this carbon fixation is really the thing that we need to know, is really important. So this is when we convert our CO2 with the use of rubisco into this molecule called 3-PGA. So this is where we say that the carbon from the inorganic CO2 is fixed into an organic compound. So this is where the main meat of it happens. So we've turned our CO2 into an organic form of carbon, which we can also call 3-PGA. There's another name for it. Then there's these two other stages as well. So we've got our reduction and then our regeneration. So in the reduction stage, this is where our cycling coenzymes are coming in. So this is where NADPH is donating its electrons to the 3-PGA that we created in carbon fixation to create this molecule called G3P, yep, which is also over here. And then we've got regeneration. So this is when the rubisco molecule starts the cycle again, and it's basically just over and over and over. It's cycling, cycling, cycling. And this is where our ATP comes in, because to continue on with this cycle over and over and over requires a lot of energy. And this is where our coenzyme of ATP comes in. So we've got our carbon fixation, turning inorganic carbon into organic carbon. Then we've got our reduction, which is where our coenzymes help to further, refine that process into the glucose molecule. And then we've got our regeneration, which is the repeat of this cycle over and over until we've got the glucose molecule that we want at the end. And this is using our coenzyme of ATP. So that's what carbon fixation is. And rubisco plays a really, really heavy part in that, because obviously it's the enzyme that converts the CO2 into the 3-PGA to create our organic glucose molecule. Now, like I said, there is problems. Rubisco's got a lot of problems. It's not the best enzyme in the world. It's definitely not my favorite enzyme. It's definitely a flawed enzyme. So rubisco is normally pretty good at this. They're normally pretty good at fixing carbon from CO2. They're normally pretty good at what they do. But sometimes, apart from being, really inefficient and slow, sometimes instead of using CO2 as a substrate like they're supposed to, sometimes it uses oxygen instead. So we know that oxygen is a product of the light-dependent stage. It might be floating around. It's not really needed, but because it's floating around, rubisco sometimes, has a wandering eye that likes to bind to oxygen instead, because it's got a really similar shape. Without rubisco binding to CO2, obviously that's a major problem, because that means photosynthesis cannot proceed. If our carbon dioxide is not being converted into glucose, there's literally no point of doing photosynthesis, because the whole point is to produce the sugar. So when rubisco binds to oxygen instead, we have a major problem. And this binding of O2 instead is called photorespiration. So photorespiration is a really big problem. It's super wasteful. It means there's less photosynthesis. We've got less glucose production. It wastes a ton of energy. So why? Why would it do such a thing? Why would it have this beautiful CO2 that it's got here, and why would it go for this O2 instead? There's two main reasons that this could occur. One of them is substrate concentration. So we know from our enzyme stuff, we know that the more substrate that's present, the more chemical reactions will happen. So if there's more oxygen substrate present, so if, there's been a lot of light-dependent reactions happening, if the plant, if there's a lot of oxygen around, there's a greater chance that it's going to bind to rubisco to undergo a reaction. Because of this, plants want to expose rubisco to high CO2 concentration. So to facilitate this, the stomata of the plant stays open to allow CO2 to enter and O2 and water to leave. So we want to bring carbon dioxide in, we want oxygen to leave. However, when a plant needs to conserve water, it will close its stomata. So, in perfect circumstances, carbon dioxide is coming in, oxygen is leaving, this photorespiration does not occur. But when the stomata close to conserve water, the oxygen that's produced during the light-dependent stage from the splitting of water builds up inside the cells. Because it's building up, that means there's a greater concentration of oxygen inside the cell, which can increase the chance of photorespiration. Because if there is lots and lots of oxygen around, and it's got a really similar shape to carbon dioxide, there's more chance that rubisco is going to bind to it. So one of the main causes of this photorespiration problem is O2 being trapped inside the cell and being bound to rubisco instead of carbon dioxide. Another reason this can happen is because of temperature. So at regular temperatures, normal temperatures for a plant, rubisco's affinity for carbon dioxide is awesome. So we know that enzymes are really affected by temperature. So if the optimal regular temperatures are around, CO2 is going to bind to rubisco. But if temperatures increase, so at higher temperatures, the affinity for oxygen is actually higher. So that leads rubisco to bind to oxygen more. So in lower temperatures, rubisco is going to bind to carbon dioxide, but if those temperatures creep up, the affinity is going to change and oxygen is actually going to have a higher affinity to rubisco than carbon dioxide is going to. So changes in temperature can also cause photorespiration to occur. So obviously this is a massive problem. If we can't produce glucose, what's the point of doing photosynthesis? Rubisco is really bad at its job, but we have adapted, well not we, plants have adapted to prevent this from happening. So remember in our last video I said that when we're talking about photosynthesis, I was talking about your traditional C3 plants, which is just your normal plants. Well, there's two other different kinds of plant that we're going to learn about now, and these two kinds of plants have evolved or adapted to prevent this photorespiration issue from happening. The first of those plants are C4 plants. Okay, so a C4 plant is an adapted kind of plant. So what they've done to prevent this photorespiration from happening is they physically separate the light-dependent and the light-independent reactions. So they physically separate them through two different types of cells. So you'll see here, the light-independent reactions occur in mesophyll cells. So they have compartmentalized, so they have kept our light-dependent reactions are only occurring in our mesophyll cells. So they are trapped. So all of the oxygen that is produced in this light-dependent reaction is also trapped in that cell. Then what they've done is that they have split up light-independent reactions into a different cell type. So they are called bundle sheath cells. So the light-dependent reactions occur in mesophyll cells, and the light-independent reactions occur in bundle sheath cells. So this does a few things. So one, this keeps oxygen trapped inside the mesophyll cells. So that means that oxygen can't build up in our bundle sheath cells, which is really awesome. So with the mesophyll cells constantly pumping a source of carbon dioxide into the bundle sheath cells, so this cell type will continuously pump CO2 into here. So we're still getting our CO2. So that's not a problem. So the mesophyll cells are going to continuously pump CO2 into our bundle sheath cells. So that means that there's always going to be a higher concentration of carbon dioxide in our bundle sheath cells. So that means that rubisco is always going to bind to carbon dioxide in the bundle sheath cells because one, it's all that's there, and two, oxygen is trapped in the mesophyll cells, so rubisco cannot bind to it. So C4 plants have separated these two reactions across different cell types to trap oxygen and carbon dioxide into two different cell types, which means that photorespiration is really, really minimized and photosynthesis is maximized. So this is what C4 plants have done. So that's what makes them really unique. They're more suited to like a higher temperatured climate. So C4 plants have adapted in, maybe tropical areas, places where the temperature is a little bit higher. Why do you think they've adapted that? So we're kind of going into evolution now. Because the temperatures are higher, there would be a higher affinity for oxygen, which means they needed to adapt really fast because those plants would not have survived if they just let photosynthesis take over, photorespiration take over, so they needed to adapt. So that's why these C4 plants that are in, higher temperature climates have adapted so well and C3 plants haven't done so well because they haven't really needed to because they're in lower temperature. So they've adapted this really cool mechanism to prevent photorespiration from happening in those higher temperatures, which I think is very nice. We then have our CAM plants. So just like C4 plants, CAM plants have also really needed to adapt it because they're from hotter environments. But the difference between C4 and CAM plants is that CAM plants are also in really dry environments. So CAM plants adaptations are a little bit different because they've tried to hit two birds with one stone because they are trying to combat photorespiration, but they're also trying to combat water loss as well. So CAM plants are things like cactuses and pineapples, things that are in those really dry, arid environments. So what the CAM plants have done, they've kept our light independent stage exactly as is. So the light dependent stage of photosynthesis in CAM plants is the exact same as C3 plants. So it's the same sunlight, water splitting, all of that sort of stuff remains the exact same. What they've changed is the light independent reactions. What they've done is they've split them over time. So C4 plants physically split them through cell types. What CAM plants have done is they've split them through time. So the light independent stage is split from nighttime to daytime. So what they've done is during the night, they open their stomata because one, it's cooler, and two, there's less chance of water loss and things like that. So at night, our CAM plants open their stomata. And when they're opening it, they're bringing in our carbon dioxide and they're completing the first stage of the Calvin cycle here. So they are completing carbon fixation at nighttime. So what does this mean? This means that because it is cooler, because there's less oxygen around, performing the carbon fixation step here means that photorespiration is very limited because it's cooler, because there's less oxygen around at nighttime. Photorespiration doesn't happen as much. So they do this at night. So they bring carbon dioxide in at nighttime. Then during the day, they have their stomata closed. This is for several reasons, but the main one is to prevent water loss because during the day it's really dry, it's really hot, so their stomata is closed. And then this is when our reduction regeneration steps of the Calvin cycle occur. So Rubisco isn't really necessarily needed in this part because Rubisco has already done its carbon fixation at nighttime, so then the rest of the process occurs during the day. So we've got our coenzymes are working, our glucose molecule is created. So that's how the CAM plants do it. They split it over time. So C4 splits it over cell type. CAM plants split it over time. So they bring in their carbon dioxide at nighttime. When it's cooler, they complete the first part of the carbon fixation process using Rubisco. And then in the day, they do all of the rest of the stuff that they need to do to produce glucose and limit photorespiration from occurring, which is excellent. So on this last slide, I've just got a summary of the different kinds of plants and their different adaptations and why they've occurred. So in this point here, we've got C3, which is our general run-of-the-mill photosynthesis, where photorespiration is more likely to happen because they've got no adaptations. So C3 plants have no adaptations to limit photorespiration, and they've got none of these adaptations that the C4 or the CAM plants have. So the disadvantage is obviously they're more susceptible to photorespiration, but they also don't consume the extra energy that the other plants do. So this is most plants when we're talking about photosynthesis. We've then got our C4 plants. So they have the adaptation of separating their carbon fixation over two different spaces. So they have the first part of carbon fixation in the mesophyll cells, and they have the second part of carbon fixation in the bundle sheath cells, so that the oxygen is trapped in the mesophyll cell type, and then the rest of carbon fixation with Rubisco can happen in the bundle sheath cells and is not having to compete with oxygen. So they split their two different stages over different cell types. This uses a lot of energy, but it also minimizes photorespiration. So they are best adapted to the hot climates like we talked about because they needed to adapt because photorespiration would be more likely in hot climates. And then we have our CAM plants. So they have separated their light independent stage over time. So what they've done is they've had the initial carbon fixation step happens at nighttime where it's a lot cooler. And then the rest of the Calvin cycle happens during the day when the stomata are closed. So this again consumes extra energy, but it also minimizes photorespiration and reduces water loss because they are often in very dry, hot habitats. So hopefully this helps. If you have any questions, please bring them to class and I will see you there.